So let's start our new panel and invite as a chair for the panel in health agenda, the T20. No. We invite Milena Botelho Pereira Soares for the panel RNA-based solutions, infinite possibilities in biotechnology. She has a degree in Harvard in the United States. Is one of the Brazilian founder of genic cellular therapy, and she's also a consultant for Anvisa in the area of cellular therapy and genetics. She coordinates the laboratory for immunopharmacology, and as speakers in this panel, we invite the manager for the RNA in Bio-Manguinhos, Patricia Neves, who have the presentation mRNA vaccines platform. She will present this presentation. She's a doctor in cellular biologist, and also she has a master in this area, and she's graduated, has a degree in Federal University of Rio de Janeiro, and she works in biotechnology in Bio-Manguinhos, where she participated in projects for developments of vaccines and biopharmaceuticals, and she works in a regional hub of development and production of RNA vaccines for the WHO, with the scope of development of vaccines and therapies based in RNA. She also integrates the coordination committee of Cancer with the programs of translational programs of Fiocruz, Senior Vice President of Alnylam Pharmaceuticals, Jing-Tao, who will make the presentation RNAi therapeutics, moving from the research of reality, and he will participate online. He's a PhD in chemistry by the Michigan University. He has worked at DuPont Pharmaceuticals, Millennium Pharmaceuticals, and Takeda Pharmaceutical Company, with increasing responsibility in preclinical developments, and he published more than 50 articles, revised articles, and he acted in several scientific conferences. I pass the floor then to Milena Botelho Pereira Soares. So good morning, you all. It's an immense pleasure being here, coordinating this panel with this such important and relevant subject, with the use of molecular molecules and RNA molecules in vaccines and therapies. We're going to see two presentations, and the one of vaccines and the other development of therapeutic products based in RNAs. And in the end, I'm going to also make a small presentation regarding all the development of RNA vaccines in Bahia. So I invite Dr. Patricia to start. Thank you, Milena. Good morning. So in the following minutes, I'm going to tell you what has been the story of the development of the first Brazilian vaccine, RNA vaccine, and the deployment of the platform in Bio-Manguinhos and Fiocruz. I'm going to end my speech showing the work the results that we have been having, and we have been seeding the innovation or regions in this area of RNA products. I would like to start talking about, I'm sorry, this is not working. The pointer is not working. So I'm going to talk about the context and how we started this project. And this image shows this map is one of the websites that publish this kind of study, and it shows it's from September 2021, and this date is very important for us, and you're going to understand better. And this map shows the situation of vaccination in the world in the peak of the pandemic in 2019. So you can observe that we have uh, countries with very high vaccine coverage. That's United States, China, Canada, countries from Europe, and especially the African countries with very low vaccine coverage. And Brazil had an average coverage due to institutes as Bio-Manguinhos and Butantan, and our situation was not as bad as Africa. So even though we were behind many other countries, more developed countries. So this was called vaccinal apartheid. So this situation called draw the attention of the WHO, and it took the World Health Organization to make this calls for expression of interest to companies and institutes that were developing COVID vaccines based on mRNA platforms, and that could make available this technology to low-income countries. In our region, this call was done together with the Pan American Health Organization. And at the time in Bio-Manguinhos, we were already working in the development of RNA vaccines for the treatment of cancer, following the portfolio of great companies like Moderna and BioNTech and other countries. We changed our focus to COVID, which was the interest at the time. We submitted this call, and with much happiness, we were congratulated with this choice of Bio-Manguinhos as one of the institute that was contemplated in Latin America for the development of this kind of production with Synergium from Argentina.
And on that day, on that map, we were announced by the Pan American Health Organization. Then it's a very important date, and it's the birthday of my partner at the project, and he got a birthday gift from the World Health Organization. We began this project working on DNA, self-replicated DNA construction projects, as you might know. The self-replicating RNA is different from the RNA, conventional RNA that's been used in current vaccines, because it relies on on genes from the proteins of some viruses, of the replicated complex of some virus, usually an alpha virus, and we began then to do a construction containing two antigens of COVID, with the goal of having a wider protection faced with the emergence of new variants. However, the self-amplifying constructs are more challenging in terms of delivery. We had some good results with our constructs, self-amplifying constructs, but we also began to work on conventional RNA constructs because of the difficulty in the delivery of self-amplified vaccines. We thus designed five-line and three-line elements that are differentiated from those that are currently used in vaccines, and that are patented structures that are different from the previous ones. And we've done several modifications in the sequencing of the spike protein, which allowed us to leave the patents that have been filed. And this is then the core of the patent that we'll be filing briefly. In this period of 2 years, starting in September 2021, we have tested over 20 combinations of lipids and monocytes and the constructs there referring to them, taking into account self-amplifying RNA constructs as well as conventional ones. And all of these combinations have been totally developed at Bio-Manguinhos by our team. And these ionized lipids, we took care to examine the patents by Moderna, which protect SM112 lipids, so there wouldn't be any questions pertaining to the intellectual property of the lipids that we're using. I cannot show you the results, the real outcomes, unfortunately, because of the patent, but some of these candidates that we have tested had excellent results. We have tested these candidates in a K18 animal model with a very strict challenge, lethal challenge that kills the animals. So we had some of these candidates that we have tested, they had a reduction of pulmonary cerebral viral load, 100% protection against the lethal challenge, and cellular responses that were robust, and cell titles of neutralizing antigens, including cell titles of neutralizing antigens bigger than our current comparator. As was said before, we have fulfilled all of the requirements, internal requirements to assess patents and skills of our constructs and formulations. We have gone through the sieve of our NITs and the sieve of our central office at Fiocruz, which is Gentech, ascertaining requirements like the inventive activity, novelty, and industrial application. We have gone through the assessment by the patent committee of Fiocruz. We are thus at the last phase to file a patent for this construct, and in tandem with the formulation that we have developed. This is a snapshot of the safety data. I can show you these data, showing you the biodistribution of our vaccine 6 hours after application and 24 hours application. 24 hours after application, this in vivo delivery test. It's a very safe vaccine, as you can tell from what I'm showing you, even though we have signals and the liver, which is common after 6 hours of application. After 24 hours, we don't have any signs in any other organ that other than the muscles. 144 hours afterwards, we still have some signs at the site of application, and 336 hours later, we don't have any signs in the animals, which shows the safety of our construct. Now talking about onto our the facilities of our production area, because in addition to the development of COVID vaccines within the scope of the project, we're completely implementing the platform and the platform at Bio-Manguinhos. As you can tell from the picture, our production area is located on the 3rd floor of that building on our campus, Fiocruz campus. The production area is virtually ready. We're waiting for the final equipment, and it will be the first RNA plant and good practices of manufacturing in Latin America. We have adopted as our production system of the RNA the quantum system of a Belgian company with which we are partners, and we have installed and tested our equipment towards producing nanoparticles in according to good manufacturing practices. I'd like to ask our technicians to show you the video of the quantum video showing its operation.
Esse é o nosso equipamento já.
This is our equipment already installed in Brazil. And this is one of the test runs that we have done in our production area. The equipment is totally operational. In 2 weeks' time, we'll be producing our first lot of drug substances in good practice, according to good manufacturing practices. It will be the first drug substance lot produced in Latin America. In terms of process development, we have many laboratories working in this process development. We have the recombinant technology working on the plasmodium. Um, uh, in the GNP area, we hope to get to at least 6 liters, and with a final yield of 540 milligrams of plasmodium was enough to do 36 rounds of in vitro transcription, which is more than enough for our demand. We also have scaled up the production of the alienization of the plasmodium. We are currently working with 20 milliliters because our current need caters to that requirement. We have the capacity to do 200 milliliters of plasmodium linearized. It's enough for 6 productions and our equipment of in vitro transcription. Our equipment of in vitro transcription will go from 200 milliliters to 800 milliliters, 200 milliliters of reaction. That's enough, sufficient to produce 4 grams of RNA, which is enough, even though the scales are similar. And our total capacity currently is 16 grams of RNA in terms of doses. It gives us 220,000 doses of 50 micrograms. These 200 milliliters of RNA can be encapsulated in 28 liters of formulation. And we have encapsulated a lot of 4 liters. We're currently on the scale of 4 liters, but for reasons of tests, it was the first test we have done virtually. We would have the ability to, for 28 liters of production, which represents in terms of doses 62,700 doses. This encapsulated RNA is formulated and filtered, which gives us a final capacity, taking into account a lot of 200 milliliters for 39,300 doses, which is more than enough to do the clinical studies. These are the data from our first lot of encapsulation and our equipment and good manufacturing practices. We have obtained a nanoparticle of 90 nanometers with homogeneity that's quite satisfactory at 0.1 reliability with a 4-liter lot, which would be tantamount to 8,000 doses, roughly speaking. And in terms of the establishment of the quality control tests, we have 70% of the tests that have already been established. We're waiting for the finalization of our production area so that we can produce viability lots, feasibility lots, and do the validation of these methods. Those that have not been tested, we already have the equipment, but it's not yet properly installed. So once we set it up, we'll go on to the development of those methods, and everything's being done in tandem with a tandem work with between the Vice Directorate of Innovations and the Vice Directorate of Quality Control personnel. We've done also preliminary studies of stability with a bench lot, a small bench lot, and where we have analyzed 6 attributes, quality attributes, intra-lot and inter-lot. We've done 3 lots, And our formulation is stable for 60 days from 2 to 8 degrees, and it's less under minus 30 degrees. It's important data that we're going to be reproducing with our first lot produced in the good practices area. As for quality assurances, we produced a total of 36 documents. We have produced 36 documents of several typologies that are required for the transfer of the process from the bench area to the pilot production area, and 87% of these documents have already been prepared or have been approved. It's a joint effort that's very large, done for the first time inside our unit, with a commitment by our team and the team of the pilot plant and the team of the Vice Directorate of Quality Control. Now, in terms of milestones, what we are doing at the present time is to wrap up the tests on the two models required by our regulatory agency. We have toxicological studies planned for June, when we're going to be wrapping up our first lot and the good practices, manufacturing practices. And our production plant is basically ready. We're waiting for 3 purification equipment for nanoparticles and other equipment too for the plasmidium DNA production. And our first lot, complete lot for drug products is expected for June 2024, in a month and a half's time. In terms of implementation of the RNA platform, not talking only about the development of the COVID vaccine, but all these competencies that we have developed over the past 2 years, not only the team that worked on the initial development, but all the process of transfer of production of documents, analysis of patents, and quality control, that has allowed us to say today that we can offer through a partnership or in services the whole foundation for the development of RNA vaccines, such as the design of the antigens that we've been working on through a partnership to begin a study using artificial intelligence to do this design. And our skeleton, our backbone for the RNA vaccine, which is what we're patenting at the present time, the development of formulations and the material for the principles proof, the production and pilot scale, non-clinical lots, and our animal experiment laboratory that's prepared for animal tests. And the scaling, the pilot plant team has been working strenuously on the scaling studies and process improvements. And briefly, momentarily, we hope to have the ability to produce clinical lots and good manufacturing practices. Obviously, the development of all of these competencies has taken us to really to act as a hub for the technological development of RNA products in Latin America. And we have begun to, we have attracted through our competencies many partners, not only in our region, but also in other regions of the world. We have drawn the interest of many partners. In Europe, we have Quantum, our partner from all along in the development. And recently, we were contacted by the Bullfarm company in Portugal. Obviously, within the arrangement of the World Health Organization, we have several commitments and the joint development efforts with Afrigen and South Africa. And we're close to signing a memorandum of understanding with the the medicine patent pool that does this mediation, and the Pan American Health Organization, that was our big supporter from the very beginning. We're discussing partnership agreements with the University of Sydney, the RNA Center of Sydney University in Australia, our local partner Synergy in Argentina, our regional partners. We're talking also to Butantan in Brazil, and also the Research Center for Cancer in Paraná, because we're advancing in formulations that are therapeutical as well, and Fiocruz Mato Grosso and the Federal University of Ceará, among several other universities and companies with which we are talking to which we're talking. I believe that that's very important to our region. I believe that it's innovative, it's a breakthrough, and I think that this is the opportunity for us to show our strength as a country, as Latin America, to showcase our ability, our capacity, not only in production, and we had showcased it before with increased capacities that have surfaced, that already existed, but which have flourished in the course of the pandemic, and they are very robust at Bio-Manguinhos and Butantan. But I think that our dream when we began this project was to poise Bio-Manguinhos and Fiocruz and our local partners as hubs for the technological development, because we're only going to reach truly the equity in the distribution of vaccines and high value added products when we're able to develop our products. And the first graph that I showed you shows you very clearly that at the moment we need a sanitary emergency, for example, we still, even though we have the productive capacity, we still need other developers to transfer their technology to us. We should develop our own capacity to be totally free and independent and self-reliant, to have our own products. I'd like to thank you immensely, the team of the technological laboratory, immunological laboratory that's working incessantly all the way, and many teams of Bio-Manguinhos involved, both in the Vice Directorate of Innovations and also Vice Directorates, many joint studies being done and many innovative works being carried out. I dare say that for the first time at our institution, perhaps for the first time in Brazil. Thank you, and I'm very proud of the work we're doing. And I'd like to thank you from the bottom of my heart, all of you who are making this happen, and turning this into a reality.
Thank you very much, Patricia, for your lecture, very inspiring lecture. I'm going to lead the Q&A session to the end. And
Jing-Tao, please. Thank you for being here.
Thank you, Milena, for the introduction. Let me share my screen. First, I apologize for I cannot be there with you all in person. I'm actually in Brazil, in Sao Paulo, but I have a pre-commitment in Sao Paulo later today. So I'm here, very excited to be here today to talk to you about a new class of innovative medicines, RNAi therapeutics, that's moving from research into reality. So I want to start with a brief history of the RNAi therapeutics. Now, the phenomenon of RNAi was first observed in 1990, where researchers found their petunia flowers turn white on overexpression of a gene which is supposed to make it more purple. Now, nobody can explain what happened until 8 years later, in 1998, Andrew Fire and Greg Mello reported potent gene silencing effect in worms. And they also elucidated for the first time the biological mechanism of RNA interference. So they call it RNA interference and RNAi. And in 2006, both of them received the Nobel Prize for this breakthrough discovery. Now, shortly after that breakthrough discovery, Alnylam Pharmaceuticals was founded. Now, after that, it took a long time for Alnylam and other companies in the field to turn a research findings into a therapeutic reality, a medicine that can reach out to patients. So I'm going to go through some of that, why it takes that much time, that it's so hard to turn in research into reality. But the good news is after 16 years of extensive effort, including numerous failures, we were able to get our very first approval for an RNAi therapeutics on Patro in 2018. And quickly following that, we have 4 additional siRNA drugs got approved, including the one that we partnered and commercialized by our partner Novartis, Cibrarava, approved in Brazil. So now, before I want to talk to you about some of the key breakthroughs in the science, how we get to turn the research into a reality, I wanted to briefly review how an RNAi therapeutics works in our body. So RNAi therapeutics, also called small interfering RNA or siRNA, it is a double-strand siRNA, typically with about 19 to 25 base pairs. Now, when it gets introduced into the cytoplasm of a cell, it will bind to a protein, an endogenous protein complex called RISC, which stands for RNA-induced silencing complex. Now, once it's loaded into the RISC, now the antisense strand will get removed. The sense strand will get removed, leaving only the antisense strand loaded into the RISC. Now, this RISC actually contains a cleavage enzyme for messenger RNAs. So now this RISC-loaded antisense strand-loaded RISC will look for mRNAs that contains a matching sequence of the antisense strand. Now, once they found that matching sequence mRNA, the RISC will perform the cleaver function to cleave that messenger RNA and shut down the protein translation. Now, once this happens, now the RISC can go back and start to look for the next mRNA molecule in the cell. So this is really also a catalytic mechanism. So the same antisense strand and the RISC can work over again to shut down mRNAs. So that's why this is also a very potent mechanism because of that. And in theory, we can silence any genes in the genome. And it's important to mention that this mechanism is actually, it's a natural mechanism. MicroRNAs in the body actually leverage this mechanism, this RISC-mediated cleavage mechanism, to regulate gene expression. So now, look at this mechanism. Sounds like it's a simple process. However, to really make this to be a real therapeutic drug for patients, we have to overcome many challenges. I'm just going to highlight some of the key breakthroughs in this process. Now, first, to understand this, let me briefly talk about some of the characteristics of siRNAs. Right, as I mentioned, siRNAs, these are double-strand oligonucleotides that chemically synthesized, typically 19 to 25 base pair in length. Now, because of this base pair and the double-strand nature, so it has a large molecular weight, around 14,000 Dalton. And because it also contains nucleotides, okay, it is highly negative charged, and it's a very hydrophilic molecule. So none of these properties actually make it more like a drug attractive as a drug-like problem, right? And specifically, the challenges are first, the unmodified siRNAs are not stable after dosing into the human body or any live animals, just because we have this ubiquitous nucleus in our body that's going to cleave this siRNAs. So the first problem to solve is the stability. Now the second, because the large size of the molecule, it will not passively diffuse or distribute it into any cells or tissues. So it needs an active delivery mechanism to deliver the siRNA inside the cells. So that's the other challenge. And also we found these unmodified siRNAs also have some immunostimulatory effects. So that's another challenge. So this is all these instead. So this is natural unmodified siRNAs doesn't have any drug-like properties. So how we address this problem is through chemical modifications. After numerous and collective effort in the field, that we look at all kinds of modifications, and we finally optimize, find, and generate a a chemical modification based on 2'-O-methyl and 2'-fluoro will provide the best stability of the siRNAs. And also, it will help able to mitigate this immune activation as well. Now, this is easy to say, but once you introduce this chemical modification, it actually increases another layer of complexity, because where do you put the 2'-O-methyl and where do you put the 2'-fluoro actually make a huge impact on the potency of the molecule. So all this need to be studied and figured out. Now, I don't have time to go to detail, but another dimension of this is we start to learn that some of the toxicities we've seen with siRNAs are actually not driven by any of the chemical modifications, but mostly based by the seed-based off-target effects. Now, at Alnylam, we have, again, reached out to a chemistry toolbox, have designed a novel chemistry templates we call ESC+. That was effectively minimize this off-target effects. So our siRNAs are not only potent, but they are also highly safe in human. So now, once we address the metabolic stability and the immunostimulatory effects, now the next challenge, as I mentioned, is the delivery, right? Now think about this delivery is not only we want the siRNA to go into the tissue that we want, but we also want it to go into the cell type that we want to go into. And not only we want it to go into the cell type, we also want it to get into the cytoplasm and load onto that risk complex I just talked to you about. So through the years of innovation, now we have worked out two mechanisms for liver delivery. One is through this lipid nanoparticle. I think Patricia just touched on that. This is the method, it's a very similar method used for delivery mRNA vaccines. Basically, it's a lipid formulation. And our first approved therapeutics, Patricia, was actually using this lipid nanoparticle formulation. Now, the second delivery method is a little bit more simpler. It's using a conjugate approach. We actually leverage this is the ASGPR, which is the acetylglycoprotein receptor that's expressed on the hepatocyte surface, as a receptor to deliver our siRNA. So for our siRNAs, we conjugate it to a GalNAc, it's acetylglycosamine. It's a sugar, basically. So an ASGPR will recognize the sugar moiety and bind it and internalize the siRNA into the hepatocytes. So this is a highly effective and simple way to deliver siRNAs into hepatocytes. And I think both delivery approaches are highly effective, but most of our late-stage products or newer products were based on this GalNAc siRNA approach, just because of the simplicity of a single chemical entity. And now, because based on this huge advancement or platform in terms of the modifications of the siRNA and solving the delivery problem for siRNA, we have developed one of the most robust pipeline in any biotech companies in our industry, right? And it's across different stages and different areas. At the end, I'm also going to briefly touch on not only liver, we are now also entering into extrahepatic space, such as CNS space. But on the right side, it's so exciting to see now through all this work, a pipeline, we have now 5 approved RNA therapeutics. That's on the market. That's helping patients with unmet medical needs. So in the next few minutes, I'm going to take just a few time to give a brief highlight some of the products that's got approved in this space. I want to start with our TTI amyloidosis franchise. So this is transthyretin mediated amyloidosis. It is a rare and progressively debilitating and fatal disease caused by misfold TTR protein that accumulates as amyloid deposits into multiple tissues. Now, there are 2 major manifestations for this disease, polyneuropathy and cardiomyopathy. Now, when this amyloid deposits mostly deposit onto nerves, it will have this causes polyneuropathy. On the other hand, if it's the mostly deposit in the heart tissues, it will cause cardiomyopathy. And we have 2 products, Patricia and Vutra, that's approved for the polyneuropathy manifestation of the disease. And because those cardiomyopathy and polyneuropathy, all caused by same disease mechanism. We have an active clinical trial ongoing to evaluate vutrisiran as a new therapy for cardiomyopathy as well. Now, here's the kind of brief therapeutic hypothesis, because the TTR protein is mostly synthesized in the liver. So what we did is we designed an siRNA molecule to target the TTR gene in the liver so that we can reduce the TTR protein production and so reduce the circulating TTR level, and thus we can prevent or clear the tissue amyloid deposits and halt, and in some cases improve this progressive disease. So as I mentioned, we have patisiran, vutrisiran approved. We are very excited. Also, we have our next generation TTR silencer, TTIs04, that's in active clinical development, and which has the potential to become the best-in-class TTI silencer because it offers the potential of once-a-year dosing regimen for this treatment. And here just an example showing the phase 3 results for vutrisiran polyneuropathy study. And here we look at the mNIS score. This is really measuring the neuropathy impairment. And this is called a modified neuropathy impairment score. And the highest score was associated with worsening disease. And as we can tell, the treatment group across between 9 months and 18 months, this clearly has shown evidence that the treatment of vutrisiran, we have slowed down and stabilized the disease compared with the placebo group. And we also observed a significant improvement compared with the placebo group on the secondary endpoints, which is about reflecting the quality of life. And the drug is also very well tolerated with a very safe safety profile. The major side effects are some of the injection site reactions, which are generally mild and transient of nature, rarely result in discontinuation of therapy. Now, because the TTR, the major function of TTR is to transport vitamin A in our body. So patients on this therapy are instructed to take vitamin A supplements. So next, I'm going to briefly touch on an example in our product. It's called lumasiran or Oxaluma. This is a drug to treat a primary hyperoxaluria type 1 or PH1. Now, we're particularly excited about this therapy because it provides really a revolutionary treatment for young kids. This is usually children with very young age or sometimes infants. Who before this approval, there was not an existing therapy. They have been get on the dialysis and the disease still can progress. And I think the pathogenesis of the disease, let me just go quickly. So show you, this is the mechanism of disease. So this disease is really caused by a defect enzyme called AGT in the liver that converts glycoxylate into soluble glycine. Now, when that enzyme is mutated and lost function, so this will cause an accumulation of oxalate, and which can precipitate a deposit in the kidney and causing kidney stones and further develop into other disease manifestations as well. So what we did is we use this lumasiran and siRNA targeted to this GO1 enzyme, the gene, which is the glycolate oxidase. So which shut down the conversion of glycolate into glycoxylate, such that reduce the production of the oxalate. And since glycolate itself is a more soluble and renally cleared compound, so this is also in our studies found this is safe to shut down the GO1 mechanism. So again, I'm just showing you an example of our phase 3 trial, the data. Now, since the disease-causing mechanism is the oxalate, so we're using urinary oxalate as our primary endpoints for this trial. And as we can see in the treatment group, showed in the blue line, there's a clear reduction of urinary oxalate compared to the placebo group in the purple. And the color. So we've seen a mean of 53% reduction. And on the maximum induction, we see up to 76% maximum induction I think more than 80% of patients on treatment reached near normalization, meaning within 1.5 of the upper limit of normal. And over 50% of patients reached complete normalization. Again, we have a very encouraging safety profile for this compound. Again, very exciting drug in our pipeline. And the last example I want to give is the givosiran that's to treat acute hepatic porphyria. Now, this is a disease that's caused potentially life-threatening attacks. This is a severe abdominal pain for patients that can also lead to chronic manifestations. And I think globally, we also estimate to have 3,000 patients. Just in the US and the EU, we have 3,000 patients. And this is the porphyria, acute hepatic porphyria. It's really a family of rare genetic disease due to the deficiency of one of the enzymes in the heme biosynthesis pathway in the liver. Right, when one of these enzymes get mutated, a loss of function, which will result in the reduction of the heme, the production of heme. So the body will try to compensate that by upregulating the really upstream enzyme called ALAS1. Let me try to get my pointer. It get the ALAS1 enzyme. This is the rate-limiting enzyme in the heme synthesis pathway. Now, the induction of the ALAS1 will cause accumulation of two neurotoxic heme intermediates, ALA and PPG. And we believe this ALA and PPG are the primary cause of all the disease manifestations. And as I mentioned, this is really a debilitating disease, potentially life-threatening, and can also cause chronic pain, and also going to have multiple comorbidities. Typically, patients with porphyria can also have hypertension, chronic kidney disease, and the liver disease. So the therapeutic hypothesis here is simple, that we design this siRNA, in this case called givosiran, to target the ALAS1 gene so that we can shut down or reduce the amount of ALAS protein so that we can reduce those neurotoxic intermediates, ALA and PPG. So here shows the phase 3 results for our givosiran trial. As we can see, patients who got on treatment with givosiran have saw a 90% reduction in the in annualized attack rate compared with the placebo group. And also we have seen a significant increase of patients who get attack-free of this disease. So this is truly life-changing therapy. This compound also has acceptable safety profile. Now we did see some in some patients some liver function increase, ALT increase, but most of this mild to moderate, and they get resolved over continued dosing. As I mentioned earlier, many of these patients with this disease also have comorbidity of liver disease. So with that, I hope I have convinced you that we have truly turned a research experiment of RNAi into the reality of a new therapeutic modality, really true class of transformative medicine based on RNAi. So I do want to close my talk with telling you some of the exciting things. Now we have great accomplishments. We have these drugs. We'll continue making the drugs. But what's next? What's coming up at Alnylam and collectively in the field? I think all the drugs I talked to you about, examples, these all target the liver, a specific gene in the liver. But we know there are many, many other diseases that's beyond the liver that we still don't have a good treatment for. So I think the next revolution in the RNAi therapeutics area will be getting into this extrahepatic tissues. For example, we know CNS, we have many of these neurodegenerative disease in the CNS space that still doesn't have a disease-modifying treatment yet, right? And liver. And again, there are still additional areas that's we haven't tapped into these opportunities. And there are many, many more in muscle, adipose, in the eye, kidney, lung, you name it. I think the vision, the passion at Alnylam is we really want to be able to deliver RNA therapeutics into every tissue in the body where there's an unmet medical need. And we think we're very much on our way to achieve this goal. I just want to show you one example. This is in the new advancement in our CNS pipeline. In this area, we developed a new delivery mechanism that we call the C16 conjugated siRNAs, showing up there. Now, and we were just last year, we were very excited to announce the very first human POC of RNAi therapeutics in CNS. Nobody else other than Alnylam has ever put an siRNA in CNS. So we did it. Not only we did it, and we successfully reached the clinical POC. I'm just showing, this is the interim results from our phase 1 clinical trial. As we can see, after single dose of this ALN APP, we've seen clear and robust and durable knockdown of the soluble APP, which is the target engagement marker. That's the target we try to knock down. We see robust knockdown just after single dose. And also, not only that, we've seen the downstream change of the amyloid beta peptides, A beta 40 and A beta 42. Now, these are the soluble amyloid peptide that they aggregate and form this amyloid deposits on the nerves. That is the causal mechanism for the Alzheimer's disease. So we're happy that we see not only we engage the target of APP, but we are starting to change some of the downstream biomarkers as well. So we still have the ongoing phase 1 trial, including the multiple dosing arm in multiple regions. And we are, in addition to Alzheimer's, the early onset Alzheimer's disease, we are also very excited to study a new indication, CAA, cerebral amyloid angiopathy. So this is another area that we're very excited to get into. So with that, I want to thank everybody. I'm going to keep you posted. I hope next time when I return, we're going to report to you many, many new advancements for RNA therapeutics into various tissues, treating many, many of these debilitating diseases. Thank you very much.
Thank you, Jing-Tao. We'll leave questions to the end. Agora eu vou passar para a minha fala.
I will start my own talk. I'd like, first of all, to thank Dr. Akira and the Bio-Manguinhos team for his great invitation, for giving me the chance to talk. My presentation, if you can show it on the screen, please. Thank you. Well, then, I would like to start off initially by presenting my Senai CIMATEC. For those who don't know, Senai CIMATEC is an institute, a Senai institute in Salvador, Bahia, and it is, as with other institutions, that we have our technical department, but it's also a university center and an important technological center. They have several competence areas, over 40 areas, and we have seen, we've been supporting them and structuring one of the most recent areas, which is the health area, and which was exactly launched during the pandemic in 2020. And that situation at that time, various initiatives were conducted by the institution to support and to help with the pandemic, and one of them was the development of one of the vaccines that the Ministry of Science and Technology and Innovation was financing and supporting. So now we had 4 vaccines that were developed at universities, and also there are other initiatives by Butantan Institute, and here we have our project, a vaccine that I'll be telling you about and what we have done throughout the development of that product. This is a vaccine made in partnership partnership with HTT. It's a Seattle company, HTT. It established a partnership not only with us, but also with in India and Korea to develop products. India through Genova was the first to have this vaccine approved in India, and we accepted the challenge to do clinical assays, phase 1 clinical assays. This vaccine has a replicon, RNA replicon. It's self-replicating RNA that amplifies the copies of this RNA. And the idea behind it is that smaller doses would be needed, since there is this process of in vivo amplification process for RNA. And using this adjuvant of HTT, which is the lion component, it's an adjuvant patented by them. It's RNA is absorbed in this particle, and this is a vaccine that surfaced as an interesting vaccine because of this technology that had not yet been used in RNA vaccines. So now we submitted it to Anvisa, and we had the phase 1 clinical assay was approved, obviously for to see mostly to look into the safety of the product and to ascertain the doses for this vaccine. And also here we have the groups. So we have 3 cohorts, 2 with 2 doses and different intervals. One of them just 1 dose. And this study was unicentric and included 88 volunteers. And there was a scaling up of doses of 1, 5, and 25 micrograms based on the experience that HTT had already had. So here, briefly, this is just to tell you about the population. We had mostly we had individuals, male individuals, and most of them were males, and we had this distribution of races, declared races. In terms of adverse events, we didn't have adverse events, grave acute adverse events, severe adverse events reported on, but we have here a distribution of the various adverse events that were reported, and local pain, fatigue, and headaches. These were the most frequent adverse effects reported. And this group, vaccinated group, obviously it was a higher incidence than the placebo group in terms of reports of adverse events. So then now we have the responses by the 3 cohorts, the cohorts 1, 2, and 3, low doses, and we saw here on the graph an increase in the number of adverse events that were reported due to the increase in doses, but there were no significant changes in the frequency between 5 and 25 micrograms. As for immunogenicity, we have run analyses without ruptures of the blindness. We have the placebo group and the vaccinal group. I should say that initially we rushed to do the study at a moment in which there were no vaccines available. So we designed it for people that had not been immunized. However, because of the delay that we had and the beginning, to our happiness, we were happy to see that we had to change protocols because we couldn't find volunteers that had not been vaccinated. Individuals were selected on the basis of the previous vaccination with coronavirus, but we saw an increase in antibodies, both neutralizing and total and neutralizers vis-a-vis the placebo, and there's an indication here for an increase in the production of gamma interferon, above all after the application of the first vaccine with the LSPOT technique. And then wrapping up, summarizing the main findings, we had few adverse events. They were light, they were minor, mostly, as I said before, local pain where the vaccine was applied, fatigue and headache, and it suggests that this vaccine is really a safe vaccine. And there's a suggestion also that it could induce a response, both cellular and tumoral response. But certainly the most important result, as Patricia highlighted before, was the training of so many young researchers and scientists to increase our capacity of response capacity to this kind of disaster, but also to develop new products. I'd like to highlight, obviously, Professor Pereira, who was the leader of the team, and all the crew, Dr. Brahma Machado, and so many other researchers and scientists that took part in the study. And also, as the result of this, the outcome is that we have incorporated a great deal of knowledge that has allowed us to begin other studies. You see here a study that's been published. You can have access to it. Vinicius Rocha has developed new constructs of RNA constructs, both through replicon and RNA1, common RNA, and we've published the study recently to show the difference between the production of antibodies vis-a-vis in vaccination with variants and or individually. Obviously, we're trying to develop other pathogens as well. We're working on developing these vaccines using this platform for advanced therapies like CAR T cells, CAR T cell therapies. Just rushing a bit now, these studies have been published also. As for the interference RNA, we're interested where we work on Chagas disease. It's one of the focuses of our research. It's a neglected disease linked to chronic Chagas cardiopathies, and we've done the mapping of microRNA, circulating microRNAs, and the model, and also in patients also with the symptomatic form and indeterminate forms. And we found some RNAs, the microRNAs that are important, that were correlated and that potentially could be used as markers of the evolution, but also as therapeutics. And we chosen the MIR21 to test this hypothesis, and we're producing. And we got the interference RNA to block the expression of the MIR21 in vitro in the in vitro model, and cardiac microblasts. It regulates MIR21 regulates fibrogenesis, and we showed it in vitro with cardiomyocytes, 5 cardiac fibroblasts, that we saw the reduction of fibrosis, as well as in the infection model with trypanosoma crucis, in which these animals were treated in the chronic phase, and we already have a great deal of inflammation and fibrosis, and we saw that this treatment with the anti-MIR21 was able to block the expression of this microRNA. As you can see here, it's very potent in this inhibition and reducing the fibrosis on the heart of these animals. So that shows you that these techniques, RNA techniques, are indeed very promising. Well, then I would like to thank all of you that supported us, especially Fiocruz-Bahia and Senai CIMATEC for all the support that they gave us, all the agencies that have financed our work, and to you for your attention. Thank you very much. Do we have time for questions? We have time for one question. Anybody from the audience would like to ask a question? Questions from the audience?
So I'll ask Jing-Tao for
It will be
a question. Jing-Tao. So the examples you gave today are all in the rare disease area. What do you see as potential for prevalent disease?
Yeah, thank you. That's a great question. I think the examples I gave are in the rare disease, but I do think RNAi has tremendous potential. In fact, one of the approved RNA therapeutics that we partner with Novartis, Leqvio in Brazil, Cibrorava, is RNA therapeutics to treat hypercholesterolemia. It's a prevalent disease. And at Alnylam, we also have a very exciting late-stage clinical development program called zilebesiran. It is targeted to treat hypertension, which is also, again, it's a huge global prevalent disease. And we were hoping that our siRNA therapeutics, zilebesiran, in this particular case, can provide its unique features. Not only that it's able to reduce the blood pressure, but also can provide this what we call tonic control of blood lowering. So what we've seen, and we have already seen this in 2 of our phase 2 trials, where zilebesiran has demonstrated a significant blood pressure reduction up to 15 millimeter mercury. It can not only reduce it, but also can reduce out throughout the whole day. So we're looking at this therapy as potentially not only improving the quantity, but also the quality of blood pressure lowering. And this has the potential for once every 6 months dosing, which can also address a lot of the issues with the adherence to medications. So we're very excited about kind of RNA therapeutics going to prevalent diseases. And I think it has tremendous potential.
Thank you. Eu fui comunicada que a gente não tem mais tempo. Thank you, Jing-Tao. Obrigada a todos. Quem tiver perguntas também para a Patricia, podem perguntar a ela aqui. Obrigada.
questions for Patricia. You can ask them later. Thank you, Milena, for your conduct, for your moderation. Thank you to Jing-Tao. And before you go to lunch, remember that at the end of today's session around 5 p.m., right after the presentation, we have Chico's women here in the Easy Arena. As I said before, we're going to break for lunch, and we have lunch boxes. And please take a minute to visit the posters, booths, and the stands, the booths outside. See you at 1:30. We'll be back here at 1:30.